Assalamu alaikum dear students. Welcome to 10 Film Technology course, lecture number 34. Be remember this is the last lecture of this course, after which the course will be almost completed. So in these sections, we will discuss the growth methods, particularly molecular beams and liquid phase epitaxy as a common deposition methods for thin film and IC fabrications. I'm Dr. Pervaiz Ahmad. So let's start today's lectures with molecular beam epitaxy first. So we remember molecular beam epitaxy, and short we write it as MBE. So the schematic diagram for the molecular beam epitaxy can be the one just like you can see it here and this particular figure. It has different far and different materials that can be utilized and this sketch you can see it here. So with the passage of time we will explain all these things that's been given in this particular sketch. So what we have, what is MBE or molecular beam epitaxy? Molecular beam epitaxy or MBE is an epitaxial process involving the reactions of one or more thermal beams of the atoms or molecules with the crystalline surface under high vacuum conditions. So you know here, this is the chamber, and remember that this chamber is being kept under ultra high vacuum conditions. I mean, this is the chamber for the process, and which is being kept under ultra high vacuum chamber. So MBE or molecular beam epitaxy is basically the process in which one or more thermal beam of the atoms or molecules with a crystalline surface under the conditions they are made to react. So one or more atoms. So here you can see that we have more atom, that is we have here you can see we have atoms, that is gallium. Similarly we have the arsenic, and we have the aluminiums. And you know that there been a coil that's been wounded around I mean the container that containing the material like aluminiums or arsenic or the dopants. Here you can see again the dopants and the gallium. So what are these the coil, what's have been these coiling being used for? So these coil that they're basically we utilize as a heating coil, because of which we're trying to heat these materials to make their I mean it's the required material, the required vapor to deposit it and the on the substrate. So this is the substrate, and above the substrate again we have heaters. This the heaters, and here we have the electrons guns that's being utilized for I mean it's for the bombardment the substrate. Here's we have the mass spectrometers. Uh, and here you can see here is liquid nitrogens, liquid nitrogen cool crab panels. So these are basically the different part of the molecular beam epitaxial system. It's basically a sketch, and we have more atoms. So more atoms, more than one atoms. I mean here you have clearly observed, and that is in this particular. I mean it's, in this particular process, we utilize three materials, known materials, or the atom, that is aluminiums, arsenics, and germaniums. And along with that we also utilize a few dopant, that is I mean dopant first and dopant second. We utilize two dopants and three of the atomic compounds are the atomic materials. So this is why we're saying that MBE is a epitaxial process involving the reaction of one or more thermal beam of atom. So here you can see that the heating coils they're being utilized. I mean, around this container that are being utilized to generate the beams of the atoms or the molecules, and to move that to the I mean to the crystalline surface under the high conditions. I mean here we generate the beams, and here you can see the beams is moving toward the substrate to be deposited in the form of the epitaxial layers. So in these techniques we have precise controls in both chemical compositions and dopant profile. I mean, whatever the materials that we use as a chemical or we use as a dopant, so all of them we have I mean the precise controls on all of them. So it has very low growth rates that, for example, for gallium arsenide, the value of the growth rate is 1 micrometer per hour. I mean, we remember it's a typical growth rate. So this is why we were saying that the growth rates in molecular beam epitaxy is very low. How much low? The example of which is given as gallium arsenide, and it's been mentioned that the gallium arsenide has a low growth rate that is equal to 1 micrometer per hour. And we remember this is a typical growth rate for gallium arsenide and MBE. Single crystal multi-layered structures with the dimensions on the other of atomic layers can be made or can be deposited with these techniques. Effusion cell or Knudsen cell is the most common type of the MB molecular beam epitaxy source, and sources of this type are sometimes called as Knudsen or K cells. I mean, this is some kind of special experimental arrangements or experimental setup that has been most commonly utilized for molecular beam epitaxy or as a source of the molecular beam epitaxy, and it is known as effusion cells or sometimes called as Knudsen cell, or for short the people call it as K cell. So the crucible and the source materials, I mean, in this kind of setup that we call a fusion cell. So in this kind of cells, we have the crucibles and the source materials that are being heated by the radiations from the resistively heated filaments. I mean, we have resistively heated filament that is being utilized to heat up the source materials and the crucibles, and we have a thermocouple in this setup. That thermocouple is used to allow closed loop feedback controls. I mean, the thermocouple again it's being utilized to allow closed loop feedback control. And later on, I mean, in this slide we will show these different parts, and now I will just explain the function of these different parts. We have crucible and source materials, and we're saying that these they are being heated by radiations from a resistively heated filaments. And there we have a thermocouples, and then thermocouple is used to allow closed loop feedback control. A typical natural cell contains crucibles. I mean, this is I mean, we're just trying to explain the natural cell and its components. So the most commonly utilized that we call typical, because it's by typical we mean the one that has been frequently utilized during the process. So it's contain crucibles, and we remember the crucible is being made of ferrolytic boron nitride, uh, quartz, tungstens, or graphite. I mean, this is the content of the crucibles that we utilize in nuts and cell. Uh, then we have a heating filaments, and we remember the heating filament is being made of metal like tantalum, and along with that we have water cooling systems and heat shields and shutters. I mean, these are uh, the main type that the nuts and cells being composed of. So this is the nuts and cell or the fusion cells. You can see it here for yourself, and all these part that's been mentioned. I mean, here they're being tagged as 1, 2, 3, 4, and 5. So what are they? Number first, I mean, you can see it here. This one. What is this? It's basically the ferrolytic boron nitride crucible. I mean, this is the crucible. I mean, this like structures. I mean, you can see here is the crucibles where we put the precursors materials during the growth. And number two, it's number two. It's been take. What is this? It's basically the resistive heater filaments. the heating filament. And we remember, as we mentioned, that heating filament is most oftenly it's been made from metal tantalum. I mean, this is the resistive heater filament, and typically it's been made from the metal tantalum. Then, and number third, we have this is the metal foil, and the metal foil, I mean, it's around these structures we have this. This is basically the metal foil denoted by 3. This is the metal foil radiation shield, and it's basically used as a heat shield and as a shutter. In number 4, just like you can see it here, this band-like structure or ring-like structures around the crucible. This is basically the thermocouple. This is the thermocouple. And what for the thermocouple is being used? A thermocouple is being used to allow closed loop feedback control. And then at number 5, here you can see it here. This is basically a mounting flange, or we can also call as a shutter. So these are the different part of the effusion cell, or Knudsen cell, or the K cell that are being utilized in molecular beam epitaxy. Then we have another structure that we call RHEED. It is also utilized in molecular beam epitaxy. Um, what basically RHEED stand for? RHEED stand for reflections, high energy electron diffractions. I mean, RHEED is basically the abbreviations for reflections, high energy electron diffractions, and it is a technique which is being used to characterize the surface of crystalline materials. Let me repeat again. Reflection, high energy electron diffractions, which in short can be written as RHEED, is a technique which is normally used to characterize the surface of crystalline materials. A RHEED system consists of an electron source that normally we call gun, and a photoluminescent detector screens. So what we do with the electron guns? So the electron guns basically generate a beam of the electrons. So what we do with that? This electron beam, I mean, it is used to strike the sample at a very small angles relative to the sample surface. I mean that you can see it here. We have this typical structures, typical sketch of the experimental setup for the RHEED. Here you can see each and everything. This is the electron gun. So what we do next? The incident electrons that we generate from the electron gun is being diffracted from the atom. I mean, here you can see that we have the sample, and this sample we have the atoms. So we say that these electrons from the electron guns, when they are made to fire on the sample, so they are being diffracted from the atom at the surface of the samples. So after that, they interfere constructively at specific angles. I mean, just like you can see it here. These are the angles, and the angle they mostly range from 1 to 2 degree, and form irregular patterns. I mean, this is the RHEED pattern. You can see it here. And these RHEED patterns in the form of image, it can look like this one. The RHEED pattern is formed here. So in the palm of screens, you can observe it look like this. So this is the particular RHEED process. You can see it here. I mean, you were saying that it's depend mostly on the electron angle. So here you can clearly observe this is the just like we mentioned, this is the screen image. So the screen image, these are the screen image for the different compounds for different material that we have deposited in the form of thin film. So here you can see that we have a monolayer growth. These are the different form of monolayer growth that are being grown by utilizing different angles of the electrons beam, and this is the form of the RHEED signal. So here this is the 001 or 1 dash 10 or 110 structures, and this is mostly formed when we strike the electron beam with an angle that is theta is equal to 0. So in that case, you can see it here. We have the substrate, and upon the substrate, I mean, just like you can even observe here, we have now atoms or layers deposited at this particular substrate. So in such conditions, we can have the RHEED signal in this particular form. This is the RHEED signals. But if we increase the angle from 0 to 0.25, so here you can see that the electron beam strike the surface, and here we have few atoms. They are being deposited at the substrate, just like you can see it here, and that can be most widely viewed here at this particular I mean monolayer growth. So here you can see that this one is completely empty, but here you can see that we have the atoms that are being deposited at different orders. If again we further proceed the angle, further increase the angle, so that is we increase the angle to 0.5 degree. So here you can see that we have more depositions of the atoms. I mean, we have more atoms, they are being deposited as substrate. In such a condition, the signal, the RHEED signal look like this. I mean, we have the atom here, just right in the middle of the first. So you can see it here. We have more deposition in the substrate. And again, if we further increase, so here you can see that we have more atom again, just like you can see it here. And if we increase it to 1, that is degree equal to 1, so again we will reach to the final conditions, which where we have almost no atom at the surface. So this is how the pattern, I mean, these are the pattern, these are the photograph of the RHEED process. I mean, that normally we have shown here, that this the screen image of the RHEED process. I mean, and how it depend upon the electron beam. So here you can see that initially we have layers 1, 2, 3, 4, and then we have the fifth layer that's been formed by just changing the electron beam angle. So here you can see that we have what we have these deposition, but here again when we increase the theta equal to 1, so here you can count the layer again. So 1, 2, 3, 4, the 4. So it's mean that we have to control the angles. I mean, at 0 we have nothing, but even if we reach to 1, so again we have nothing. So all the electrons, they strike the surface, they just move away from the surface. So the 3D surface results in a spotty patterns due to the electron transmissions through the surface roughness. The more 2D surface giving rise to commonly observed RHEED streak, and that we have already discussed. So the typical photograph of the molecular beam epitaxy system, you can see it here. This is the typical structure, the typical photograph of the molecular beam epitaxy. So here you can see that this is the deposition chamber. This is the shatter, and these are basically the RHEED screens where you can observe the depositions. This is the beam source, the preparations chamber. This is, I mean, the big drum like structure. This is the preparation chamber, and here this particular part is called the castle entry lock. And this portions is basically the analytical, we have the analytical facility, that is an XPS, outer electron spectroscopy, or X-ray photoelectron spectroscopy. And this kind of structure is only available in some well-equipped lab and more advanced countries where we have the university. They have I mean the apparatus for the growth of thin film for IC publications. Liquid phase epitaxy, that in short we can write LPE. The experimental setup for the LPE crystal growth is shown like this ones. In this we have the thermocouple, graphite sliders, gallium arsenide substrate, galliums. I mean, here you can see that, and these structures, you can see it here. We have galliums that melt with the arsenic, aluminiums, and dopants. So this is a typical structures for the liquid phase epitaxy. So what happens what happened in LFE or liquid phase epitaxy? So liquid phase epitaxy involves the growth of epitaxial layers on crystalline substrates by direct precipitations from the liquid phase. That means we have the raw material, the precursor, and the liquid phase. So this process involves basically the growth. I mean, we grow the epitaxial layers, and for that epitaxial layer growth we utilize crystalline substrate, and the growth happens from the precursor in the liquid phase, and it results by direct precipitations. I mean, the direct precipitations from the liquid phase on a crystalline substrate, we can get epitaxial layers via the liquid phase epitaxials, liquid phase epitaxy techniques. And alpha, the substrate itself in contact with the supersaturated solutions, that is arsenic saturated solutions of gallium. That is, we have it here. This is gallium arsenide substrate, and we have gallium melt with the arsenic. So here we are saying that in LPE, the substrate is held in contact with the supersaturated solution, that is argon saturated solutions of gallium. So cooling the arsenic saturated solution of gallium causes the arsenic to precipitate in the form of gallium arsenides. So the typical condition rates for monocrystalline film range from 0.1 to 1 micrometer per minute. I mean, this is the typical growth rates. We remember the LFE is the LFE in most cases is a very economical deposition technique, especially when upscale to mass production is needed or required. So that's all we have for this lecture. Thanks for watching, and this was the last lecture of this course. Now the thin film technology course is almost been completed. We remember we have skipped some of the process. We have skipped the mathematical discussions, and we have also skipped some of the calculations, which we think that it shouldn't be the part of this course, because most of the students that we have, they're working in lab with the experimental procedures. So normally I mean, so when you are an experimentalist, so sometimes the calculations that might be problems for the experimentalists. So that's why in these conditions we have skipped the majority of the calculations. So we have just given the things or describes the activities which been involved during the experiments, and so we have avoided the calculations. So that's all we have. I think you people might have enjoyed the course. If you have any query regarding the questions, I mean, you can email or you can call, and that we can discuss again if needed. So thanks for watching once again. See you again with some more physics and other lectures. So till then, bye bye.